Everyone, welcome back to my channel. Today we'll be covering Pearson Edexcel Biology Paper 2B from June 2023. So let's get started. Question number 1 states, read the passage below, use the information in the passage and your own knowledge to answer the questions that follow. So this passage is pretty long and I personally prefer to go directly to the question and then later come back to the passage. So let's start with the questions. A part, which of these processes produces ATP? So the process is respiration. Next up we have explain why intensive farming and deforestation would cause an increase in populations of dinoflagellates, lines 9 to 11. So let's go back to lines 9 to 11. Okay, so this paragraph, to generate light, dinoflagellates use special proteins and the ATP produced within their cells. Many species of dinoflagellate contain chlorophyll and are able to photosynthesize. The appearance of glowing dinoflagellates in the sea used to be a rare event, but this now occurs much more frequently. Many of the events occur in the sea, around river estuaries, and scientists think that intensive farming and deforestation could be to blame. Alright, so now they have mentioned about that they photosynthesize, they contain chlorophyll, and that they are near rivers. So the points that we'll mention over here are more carbon dioxide for photosynthesis, moreover fertilizers from intensive farming, and increased magnesium for chlorophyll and nitrates for amino acids. Moreover, we can also talk about leaching and soil erosion. So these are around 1, 2, 3, 4, 5 points. So you can talk about any 3. Next up we have explained why oxygen levels decrease after a series of glowing events. So what happens is decomposition by bacteria, or you can even say respiration by microorganisms. Next part, which type of bacteria converts ammonia into nitrates? So the bacteria is nitrifying bacteria. D part, each of the 15 copepods ate glowing dinoflagellates at a mean rate of 40 dinoflagellates per hour. Calculate the mean rate at which each copepod ate dinoflagellates that were not glowing, lines 18 to 21. Alright, so lines 18 to 21. When the dinoflagellates glowed, the copepods ate 1200 dinoflagellates in 2 hours, and when they did not glow, they ate 2100 dinoflagellates in 2 hours. Alright, so in the question we are talking about the dinoflagellates that did not glow, which were 2100. So we'll first divide 2100 by 15, which will be 2100 by 15. The answer to this will be 140. Then we'll multiply 140 with, we'll divide 140 with 30. So the answer that we'll get will be 4.67, and then finally we'll multiply this answer with 15 to get our final answer, which is 70. Okay, so 70 is our final answer. Next part, explain how natural selection could have resulted in the evolution of dinoflagellates that did not glow. Alright, so we'll explain the process of natural selection, which is due to mutation there is variation, and since there is variation, what happen will be glowing dinoflagellates will survive, or you can say they will not be eaten. They will reproduce and pass on the gene for glowing onto the offspring. So this is our final answer. Next part and the last part for this question is explain why using dinoflagellates for street lighting would help to reduce pollution. Lines 22 to 26. Alright, so let's go back to line 22. Some scientists think that we could make use of the dinoflagellates to provide sustainable street lighting. Tanks of dinoflagellates could be placed on top of lampposts. The dinoflagellates will photosynthesize during the day when it is light, and a stator powered by a small battery would then move them at night so they would glow. And these sustainable lamps could be carbon neutral and help to reduce pollution. Alright, so we'll mention over here photosynthesis, then takes in carbon dioxide, so less carbon dioxide is released into the atmosphere. Less use of fossil fuels, so less global warming and less sulfur dioxide, or you can say less acid rain. So these are 5 main points. You can mention any 3 of them. So with that, we're done with question number 1. Let's move to question number 2. A student uses this method to investigate water uptake and water loss by a plant shoot. Pour 100 cm cube of water into a measuring cylinder. Place a plant shoot into a measuring cylinder. Cover the surface of the water with oil. Place the measuring cylinder and plant shoot on a balance and record the total mass. Shine light on the plant shoot using a lamp. Record the volume of the water in the measuring cylinder after 4 days and after 8 days. And then record the total mass of the measuring cylinder and the plant shoot after 4 days and after 8 days. Part A states the reason for using the oil. So the reason is to prevent evaporation from the surface of water. Okay, so B part states the table shows the student's results. The volume of water taken up by the plant shoot is equal to the change in the volume of water in the measuring cylinder. This is called the water uptake. Calculate in centimeter cube per day the mean rate of water uptake by the plant shoot during the 8 days. So first of all, what we'll do is we'll find the change in the volume of water in the measuring cylinder over 8 days. So if you can see over here the table, the difference between day 1 and day 4 is 25. 100 minus 75 is 25, and the difference between day 4 and 8 is 10. So the total difference will be difference 25 plus 10, which is 35, and then this is over 8 days, so we need per day. So per day is equal to 35 divided by 8, since there are 8 days, and the final answer that we'll get is 4.375, or you can say 4.38. So this is our final answer. Moving forward to the next part, comment on the changes in the total mass of the measuring cylinder and plant shoot compared with the changes in the volume of water in the measuring cylinder. Alright, so changes in the total mass of measuring cylinder and changes in the volume of water in the measuring cylinder. Alright, so as you can see over here in the table, both volume and mass decreases, and the volume changes more time than mass. And you can see that water loss between day 4 and day 8, they are same, and then we can mention the reasons that why this has happened. Since the question says comment, so we have to mention that too. So let's mention both volume and mass decreases. Volume changes more than the mass. Water loss and uptake are same between 4 and 8 days. And then we can mention the reasons. Transpiration occurs. Some water is stored in the cells, or some water is retained, and this water is used for photosynthesis. So this is the final answer. You can mention any 4 of these and you'll get the marks. Next part states, explain why the rate of water loss would be different if a working fan is placed in front of a plant shoot. Alright, so we know that if there is working fan, that means wind blows, so there will be more transpiration. So we can mention more transpiration or more evaporation as wind blows, or you can say it removes still air and hence increases concentration gradient. So with that, we are done with question number 2. Let's move to question number 3. The diagram shows part of a human nephron. Which structure is the proximal convoluted tubule? So we know that the structure is A. Next up, we have the hollow space inside a blood vessel is called the lumen. The blood vessel labeled X as a lumen with a radius of 100. Use this formula to calculate the cross-sectional area in millimeter square of the lumen of this blood vessel. So we have been given the formula, we have given the radius and everything, so we just need to put the values and get the answer. But we need to be careful between the units. So first of all, we need to convert 100 into millimeter by dividing 100 by 1000, which will be 100 divided by 1000. Let's write it. 100 divided by 1000, which will be 0.1, and then we'll use the formula, which is pi into radius square. So we'll just use this formula, pi into radius square, which is 0.1 square. So just solve it, which will be pi into 0.1 square, and the final answer that we'll get is 0.03. 0.03. Sorry, 0.03. With that, we are done with this part. Let's move to the next one. The lumen of blood vessel X is wider than lumen of blood vessel Y. Explain why this difference in the width of the lumen of the two blood vessels is important for kidney function. Alright, so blood vessel X and blood vessel Y. Okay. So we need to mention over here about the pressure. It increases the pressure for ultrafiltration so that plasma leaves glomerulus and enter and enter Broman's capsule. Broman's capsule. With that, we are done with this part. C part states protein is often found in the urine of people who have high blood pressure. Describe how urine could be tested for protein. Alright, so we need to mention the test for the protein, which is using a Biuret solution. Biuret solution. And it turns lilac or purple in the presence of protein. Okay, so we are done with question number 3. Let's move to question number 4. The diagram shows a cross section through part of a plant leaf. What is the name of the cell labeled P? Alright, so labeled P, so that is the guard cell. B part, explain how part Q is adapted for photosynthesis in the leaf. Alright, so part Q is, as you can see, the spongy mesophyll layer. So first of all, you have to identify spongy mesophyll layer. Spongy mesophyll layer. So spongy mesophyll layer has air spaces. So you can mention that air spaces for diffusion. So carbon dioxide moves in to the palisade cells and oxygen moves out. And they also have xylem to transport water. So these are around 5 points, so you can mention any 3 of them. Next up, we have a student uses this method to investigate the effect of temperature on the rate of gas exchange in leaves. Pour 100 cm cube of hydrogen carbonate indicator into each of 6 test tubes. Hang a leaf in 5 of the test tubes. Place a bung in each test tube. Place the 5 tubes with leaves into separate water baths at temperatures of 15 degrees Celsius, 20 degrees Celsius, 25, 30, 35 and 40 degrees Celsius. Place a tube with no leaf in water bath at 25 degrees Celsius. Place all the tubes in bright sunlight and record the time taken for each of the hydrogen carbonate indicator solutions to change from orange to red. The student repeats the experiment 2 more times. The diagram shows one of the tubes with the leaf. State the independent variable. Alright, so independent variable is the variable that we can change, which is the temperature of the water bath. Next up, we have the species and size of leaf were the same in each tube. Give a reason for controlling one other named factor. So you can mention light, you can mention volume and concentration of hydrogen carbonate indicator, you can mention the size of tube, pH, age of leaf, or starting concentration of carbon dioxide. You can mention any 2 of them and reason with that. So let's mention light, and the reason is it changes stomatal opening and that affects photosynthesis. So next part states, state the function of the tube with no leaf. So the function of the tube with no leaf is the control experiment. To compare and show the effect of temperature on indicator. Okay, so C part states the table shows the students' results. Calculate the mean time taken for the indicator to change from orange to red at 25 degrees Celsius. Give your answer to 2 significant figures. Alright, so 25 degrees Celsius, you need to calculate the mean. time taken. So the mean is calculated by adding all the values and divided by the number of values. So it will be 25 plus 30 plus 25 divided by 3, and the answer that we'll get is 27. After rounding to two significant figures, we'll get this 27. Next part states, explain the effect of increasing the temperature on the mean time taken for the indicator to change from orange to red. Alright, so when we increase the temperature, what will happen? Okay, so we'll mention faster photosynthesis, more carbon dioxide is removed, or you can say absorbed, increased collisions because of more kinetic energy of particles, and it levels off later as another factor limits it. That we are done with this part. Last part states, another experiment. The student places a test tube containing a leaf and hydrogen carbonate indicator in the 25 degrees Celsius water bath. The student then places all the separators in the dark for 1 hour. Explain why the indicator solution changes from orange to yellow. Alright, so respiration is faster than photosynthesis. So carbon dioxide increases in indicator. With that, we are done with question 4. Question number 5 states, hormones control the menstrual cycle. Name the gland that produces FSH. So the gland is pituitary gland. Pituitary gland. Next part, the diagram shows the changes in 4 hormones during the human menstrual cycle. Which lines represents the hormone progesterone? So the hormone is D. Next part, describe the roles of FSH and LH in the menstrual cycle. So FSH matures the eggs, or the follicles. You can mention any of them, eggs or follicles. And it also, since it has 4 marks, so you need to mention the 2 functions of each. It also stimulates estrogen. Next up we have LH. So LH induces ovulation and it stimulates progesterone. B part, reproductive hormones can be used as contraceptives to prevent pregnancy. The table gives information about 3 different methods of hormonal contraception. Discuss the advantages and disadvantages of the 3 methods of contraception shown in the table. Alright, so as you can see here that the tablets, they are less effective as compared to the rest of them, and this is the overall evaluation. And talking about them specifically, you can see the tablets, they are easy to use. You don't need any medical treatment for that, no need of doctor, and even easy to stop if you want to, if you change your mind. So this is advantage. However, tablets can be easily forgotten. So let's mention the points here. Tablets are less effective. Then tablets are easy to use, no need of doctor. Tablets are easy to stop if change mind. Next up, we have tablets are easy to forget. This is a disadvantage. You need to take it every day, as the table says that they are taken every day at the same time, so people can forget taking that. Next up, we have talking about the injection method. Injections last longer, as the table states that they need to be repeated every 12 weeks, so they last longer time. You do not need to keep going to the doctor, and you can also mention that you don't need to take it every day. You don't need to take injections every day, and you cannot forget to take. However, injections need to be given by the doctor, so that's a disadvantage. Injections can be painful. And talking about the implantation method, so implant lasts longest time. They last longest time, and you don't need to take them every day. You don't need to take implants every day. Just a minute. Okay, and talking about the disadvantage of implants, you can say implants, again, they need to be given by the doctor, and implants can also cause infection. And yeah, so these are a lot of points. You can mention any one advantage of injection, implant, and the method, and one disadvantage, and the overall one that tablets are less effective, and you'll get all the marks. With that, we're done with this question. Question number 6 says the photograph shows a plant called a snapdragon. Selective breeding has been used to produce snapdragons with brightly colored flowers. This type of selective breeding can produce snapdragon plant with brightly colored flowers. Alright, so you need to mention the process of selective breeding, which is you need to select plants with bright flowers and breed them, and then you need to select offspring with bright flowers, and then you need to breed them. Next part, micropropagation is often used to make copies of a snapdragon plant. Describe the process of micropropagation. So take small samples sterilize them with disinfectant, then place them into the agar solution. ठीक है, provide them with nutrients like glucose, carbohydrates, amino acids, etc. And yeah, this is the process. You can mention this. B part, scientists investigate the effect of exposing snapdragon plants to increasing amounts of ionizing radiation. This is the scientist's method. Take a snapdragon plant and use micropropagation to produce many explants. Expose groups of explants to different amounts of ionizing radiation. Grow the explants into the plants and record the number of differences in their phenotypes compared with the original plant. Then take samples of each of the plants and measure the number of differences in DNA nucleotides of each plant compared with the original plant. Give the reason why micropropagation is used to produce the plants to be tested. So micropropagation causes them to have the same genetic material, so all plants will be genetically identical. Next up we have what term is given to all the DNA in an organism. So the term is genome. The third part and the last part and the last part and the last question for this paper is the graph shows the scientist results discuss the effects that increasing the amount of ionizing radiation has on the snapdragons. Okay, so as you can see that more radiation increases the number of changes in DNA, and that can be because of mutations. So let's mention the points. More radiation increases the number of changes in DNA. Mutations have occurred in the DNA. Mutations change amino acid sequences. ठीक है, and this codes for a different protein. Alright, so this part says the graph shows the scientist results discuss the effects that increasing the amount of ionizing radiation has on the snapdragons. So if you analyze the graph, as the amount of ionizing radiation increases, the number of changes in DNA increases. So let's mention the points. More radiation increases the number of changes in DNA. Mutations have occurred in the DNA. Mutations change amino acid sequences and codes for a different protein. Then changes in DNA increases more than the numbers and the number of different phenotypes. And some DNA changes have no effect, they are silent. And then DNA changes may be recessive. With that, we are done with question number 6, and this marks the end of paper 2 of June 2023. I hope this session was beneficial for all of you. If you have any questions, please feel free to drop them down into the comment section, and please like, subscribe and share this video. Thank you so much. Take care.